A flower in full bloom is a biological advertisement, but the display is only the opening act. The actual objective is successful fertilization. This creates an immediate anatomical problem. The vast majority of flowering plants are hermaphrodites. They carry both the male pollen-producing anthers and the female receptive stigmas inside the exact same flower. Because of this proximity, it is highly probable that a flower's pollen will simply fall directly onto its own stigma. This chart maps the effects of continued self-pollination across multiple generations. When a plant fertilizes itself over and over, it triggers a condition called inbreeding depression. The lineage degrades. Successive generations lose vitality, and their probability of survival drops sharply. Animals can physically migrate to find genetically distinct mates. Plants are rooted to the ground. To survive, they have to protect themselves from their own biology. Over time, flowering plants evolved active structural defenses specifically designed to block their own pollen. Botanists call these defenses outbreeding devices. They are evolutionary workarounds that discourage self-pollination and force the plant to seek pollen from an outside source. This timeline shows temporal separation. Pollen release and stigma receptivity never line up. Either pollen drops early or the stigma matures first. Other species rely on physical distance. The anther and stigma grow at different positions. Pollen falls, but the spatial gap makes hitting the same flower impossible. Some plants abandon the hermaphrodite structure entirely, opting instead to grow exclusively unisexual flowers. This diagram shows a monoecious plant. Separate male and female flowers share the same stalk, allowing pollen to travel between them. To eliminate this, plants like papaya practice Dioecious. The entire plant is exclusively male or female, forcing pollen to cross an open gap to a separate stalk. By manipulating time, space, and anatomy, these structural barriers act as the first line of defense to force genetic diversity. But physical barriers have limits. Wind currents and foraging insects operate randomly, and they regularly drop incompatible or self-pollen directly onto a receptive stigma. When that happens, the plant relies on self-incompatibility. The pistil acts as a genetic checkpoint, actively vetting every single grain of pollen that lands on it. This diagram models the continuous biochemical dialogue. Chemical components on the pollen test for a structural match with pistil receptors. If it's self-pollen or the wrong species, the pistil rejects it, leaving it inert. When components align perfectly, the pistil verifies compatibility, activating the grain. The activated pollen grain immediately germinates. It pushes its internal contents out through a small opening called a germ pore, forming a new pollen tube. This tube acts like a drill. It grows aggressively, tunneling deep through dense cellular tissues of the stigma and style, heading straight for the ovary. The tube functions as a protected transport tunnel. It carries two male gametes directly to the ovule, entering through a small opening called the micropyle. At the very end of this journey, structures within the synergids called the Filiform Apparatus take over. They act as a chemical guide, steering the exact tip of the pollen tube to its final destination. Fertilization requires surviving a highly regulated biological gauntlet. Only the exact right pollen makes it through. Plant breeders need to bypass these strict biological rules. To create commercially superior crops, they have to combine desirable traits from completely different species. This is done through artificial hybridization. The breeder takes over the vetting process, forcing a specific interaction between chosen pollen and a chosen pistil, while blocking all natural contamination. The first step is emasculation. If the female parent has bisexual flowers, the breeder uses forceps to manually remove the anthers from the flower bud long before they can mature and release any self-pollen. Immediately after emasculation, the flower goes through a process called bagging. The exposed stigma is covered with a butter paper bag, creating a physical shield against any random, wind-blown pollen. Once that protected stigma becomes fully receptive, the breeder removes the bag and manually dusts it with the superior pollen they selected. The flower is rebagged, and the hybrid fruit is left to develop safely. If the target plant already produces unisexual female flowers, the breeder skips the emasculation step completely and moves straight to bagging the buds before they open. By hijacking the chemical dialogue of pollen and pistils, scientists actively engineer the yield and security of the global food supply.